Greetings, and welcome to the BRCA Research Update looking back at 2016 and plans for the future. At this time, all participants are in a listen-only mode. If you have not already done so, please close all of the programs on your computer. You may submit a question via the web by any time by using the Ask a Question feature on the left side of your screen. If anyone should require technical assistance during the conference, please click the question mark icon located in the upper right-hand corner of your screen. It is now my pleasure to introduce Samantha Deegan, Social Media Coordinator. Thank you. Please begin.
Thank you for joining us for today's webinar. The Bassler Center for BRCA at Penn Medicine's Abramson Cancer Center is the first and only comprehensive center solely devoted to funding research across the globe, educating providers and patients, and advancing care for individuals with BRCA1 and BRCA2 gene mutations. Dr. Edward and Merry Prostic have generously established this webinar as part of the Elizabeth Prostic Memorial Outreach Program in memory of their daughter. The following video is a tribute to Elizabeth, her incredible life, and her dream of creating communities that increase awareness about BRCA and metastatic cancers. The Bassler Center is proud to be a part of this mission by providing life-saving information and hope to individuals and families worldwide. Please begin the video.
Tomorrow, bet your bottom dollar that tomorrow there'll be sun.
Lizzie Prostic was an amazing young woman who believed in the promise of tomorrow. And though her tomorrows were cut short, we can celebrate and sustain her optimism. Lizzie lived large. She loved the stage and brought it to life with her voice and her energy as a talented child and as she grew into a woman. Her world was full of loving friends and family, and success in class, in sports, with girls, and with boys. Sophomore year of college, Lizzie met Michael Lundblad, a liberal and an English major. He got a preview of her leanings when he saw her quizzing a friend about senators. But she learned to love camping. He learned about couture. And political opposites, Michael and Lizzie spent 10 years together, marrying their minds and their passions. Michael loved her boldness and her softness, her interest in so many different types of people, and ability to remember the small details of their lives. The time she made to talk with her grandmother, her pearls, her red shoes that said she was not afraid to be noticed. She was not afraid. Lizzie's intellect and drive were rewarded with exciting opportunities. She blazed through the University of Pennsylvania and into the halls of government, determined to win in every venue and for every cause that was important to her. Her work for senators and on committees quickly made her a Washington insider. She rose to Chief Privacy Officer and Senior Advisor to the Secretary of Commerce before being lured away to the private sector. Not yet a lawyer, she was named Managing Director at Sonnenschein, Nath, and Rosenthal in Washington, D.C., and was completing her law degree at night at George Washington University when she died. When Lizzie was awarded a posthumous degree at the law school graduation, the entire audience stood in tribute to her spirit. Lizzie was 31 when she was diagnosed with stage 4 metastatic breast cancer, and her daughter Harper was 4 months old. Michael did research. Lizzie didn't want to know the odds, expecting to beat them. But the cruel reality was that although breast cancer is generally curable, metastatic breast cancer is not. And they found little support to help them deal with the psychological and emotional realities they had to face. In her last months, Lizzie and her family began to envision an online support network that would show patients with metastatic disease that regardless of how long they have to live, they have the ability to control how they live. This was Lizzie's final cause, and one which will be honored. Lizzie wanted to see her daughter's first birthday. She didn't make it, but she lives on in the people who loved her, and through grants such as this one being made in her name. And in truth, none of us knows how long we'll live, so we all should listen to Lizzie and make the most of every tomorrow. Live large, live strong, and wear red shoes.
Today's program will look at advances made in BRCA-related research over the past year and will highlight important work that will be ongoing in 2017. As always, this presentation will be archived and available for viewing later this week. I'd like to introduce our speakers. Dr. Roger Greenberg is the Director of Basic Science at the Vassar Center for BRCA and an Associate Professor of Cancer Biology at the University of Pennsylvania. He is an Associate Investigator in the Abramson Family Cancer Research Institute in the Perelman School of Medicine at the University of Pennsylvania and co-program leader of the Abramson Cancer Center Breast Cancer Program. Dr. Greenberg's laboratory investigates basic mechanisms of genome integrity maintenance. His research program has a particular focus on BRCA-dependent DNA repair and how this affects breast and ovarian cancer etiology and response to therapy. Dr. Susan Domchek is a board-certified medical oncologist, Vassar Professor in Oncology at the Perelman School of Medicine at the University of Pennsylvania, Executive Director of the Vassar Center for BRCA, Director of the Marianne and Robert MacDonald Cancer Risk Evaluation Center at the Abramson Cancer Center, and a Senior Fellow at the Leonard Davis Institute of Health Economics. Dr. Domchek has committed herself to pursuing research related to genetic susceptibility to breast and ovarian cancer, particularly with regard to risk assessment, screening, prevention, and treatment. She is especially interested in the development of targeted therapies in this population, as well as the long-term impact of risk-reducing interventions. And Dr. Domchek, the floor is yours.
Thank you so much, and thank you all for the opportunity for us to share with you some of the work that's been going on at the Vassar Center. We're going to start with more of a clinical focus and then segue into Dr. Greenberg's presentation with more of the basic science information. I think you all know, but there's an opportunity to ask questions, and we'll be keeping track of the questions that are asked and trying to answer those along the way, and if not, we'll try to answer them at the end. So basically, we're going to be talking about our research programs both within the institution at the University of Pennsylvania, but also work that's being funded by the Vassar Center at organizations across the country. So I'm going to start with some of the advances in cancer therapy, and we've really made great strides in this. In fact, one of the 8 milestones of 2016 in the war on cancer was PARP inhibitors in their effectiveness on ovarian cancer. So I'm going to be talking about this a little more, but I wanted to point out that BRCA1 and BRCA2 were first identified in 1994 and 1995. And at that time, we didn't even know what these genes did, what the protein's function in the cell was. Since that time, because of the basic science that has been done by people like Dr. Greenberg and others, we have a deeper understanding of how these genes and proteins work in the cell. And what that means is that once you understand how they function, you can start to decide how to target them, how to use them as the Achilles heel in the cancer. So the therapy that people are hearing most about is something called PARP inhibitors. And these are, again, this promising class of drugs for BRCA-related cancers. And the way to think about it is that PARP1 and PARP2 are proteins that help repair damaged DNA. And if you inhibit that DNA damage if you inhibit these PARP proteins, you create more DNA damage in the cells. If you have cells that don't have a BRCA1 or 2 mutation, your cells tend to do pretty well. But in a cell that already has a BRCA1 or 2 mutation and is already compromised, these PARP inhibitors create a lot of trouble and the cells die. And Dr. Greenberg's going to show you a little bit more about that later. But I want to first show you that how important these drugs have been, which is these drugs were first figured out a little bit how they're related to BRCA1 and 2 in 2005 in test tubes. They quickly entered clinical trials, and the first PARP inhibitor, a drug called olaparib, was approved by the Food and Drug Administration in 2014 for recurrent BRCA1 and 2 related ovarian cancer. A second PARP inhibitor called rucaparib was approved by the FDA in 2015. And so we now have 2 drugs that are specifically approved in BRCA1 and BRCA2 related cancers, and that's within 20 years of actually figuring out that these genes were associated with breast and ovarian cancer. So it was actually a pretty exciting time, particularly because BRCA1 and 2, because they are lost when cancer develops, they were harder to figure out how to target. And we were very excited because we were involved in these studies, and one of the studies that we led looked at olaparib, which again is one of these PARP inhibitors, and it looked at BRCA1 and 2 related cancers of any type. So the only eligibility criteria for this study was that an individual had been born with one bad copy of BRCA1 or 2 and then had either a cancer of any site. Most of the people in the study had breast cancer, ovarian cancer, pancreatic cancer, or prostate cancer, and it was largely based on this study that the initial approval occurred in the ovarian cancer patients. Now, I always like to emphasize that this approval, meaning the way that this drug can be used now for a doctor to write a prescription just in the office, is for people who have advanced cancers. So this isn't for early stage cancers, but for people with advanced cancers. We are doing additional studies to figure out when the right time is to use it, but right now there is a drug available for patients. Now, it's not curing anybody right now. These are patients with advanced cancer, but this is one more drug in the toolbox to use. And as we figure out more about it, we hope that it leads to more and more cures. The second drug, called rucaparib, it was the second PARP inhibitor that was approved by the FDA. And in this study, they studied a slightly different patient population that had received less treatment, but in this study, about half of the patients had their tumors shrink at least partially, if not completely. Again, these were BRCA1 and 2 related ovarian cancers. So similar to olaparib, this is not first-line therapy, not for early-stage disease, but still, now we've got 2 tools in the toolbox. What's important is that there was also some evidence of a signal in pancreatic cancer. So pancreatic cancer, as you may well know, is a very difficult cancer, and the risk of pancreatic cancer is elevated in BRCA1 and 2 mutation carriers. In BRCA2 mutation carriers, the lifetime risk can be in the 5 to 7% range. For BRCA1 mutation carriers, it's more like in the 2 to 3% range, and those are both compared to a 1 to 2% risk in the general population. Pancreatic cancer has a limited number of drugs that we have approved to use to treat this type of cancer, and for that reason, new therapies are particularly in demand. So in this particular study, again, led by myself at Penn, in a limited number of patients who had pancreatic cancer and BRCA1 and 2 mutations, we saw a 22% response rate, meaning in 22% of the patients the tumor shrank. That may not seem like a high number, but these are individuals who had already received a lot of therapy for their pancreatic cancer and really had no other options. So this was a signal, and we have some promising results from this study and also a second study looking at rucaparib. So studies are ongoing, and we're very hopeful that this will be a new avenue for pancreatic cancer someday. Now, we're also looking at a lot of other ways to use these drugs. So not to forget about breast cancer. There are multiple studies underway looking at several of the different PARP inhibitors in metastatic breast cancer. So these are women who have BRCA1 and 2 breast cancer that spread outside the breast. And one of the larger studies comparing olaparib to what we call standard of care chemotherapy. So in this study, women were randomly assigned to either get the olaparib, the PARP inhibitor, or a standard chemotherapy drug. And that study is closed to accrual. We've enrolled all the patients, and actually we should be hearing those results sometime within the next month or so. So you may hear a lot more about that soon. In addition, we're looking at studies such as olaparib with other drugs in combination. PD-L1 is what we call a checkpoint inhibitor, and it stimulates the immune system. You may have heard a lot about using the immune system to fight and treat cancers, and there have been some spectacular successes in certain types of lung cancer and melanoma. And the question is, can we apply this powerful tool in combination with some of these targeted drugs to BRCA1 and 2 related cancers? So it's too early to know how effective that is, but we are continuing to try to push the envelope forward and look at new ways to target these cancers. In addition, we look at other drugs. So this is another drug called lurbinectin. It also induces DNA damage repair. It's derived from a sea slug or a sea-like creature. I can't remember the details, but this is a originally naturally derived but now synthetic product. And it turns out that this too is active in BRCA1 and 2 related cancers, and seemed in this initial small study is particularly active in the BRCA2 subgroup. So again, this was another study that we've been involved in, and we hope for future work to figure out when all these drugs may be able to be used best to help patients do as well as possible. Now, how we test drugs in general is that we tend to test them initially in individuals who have cancer that's spread outside the breast or has recurred after an ovarian cancer diagnosis. So we test it in patients who've already received a lot of therapy. The question always is, if we bring it into an earlier setting, can we prevent disease from ever coming back in the first place? And we have a term for this called adjuvant therapy. And adjuvant therapy, particularly related to breast cancer, early stage breast cancer, where most women are diagnosed when they've had cancer just in the breast or maybe the breast and the lymph nodes. And we give those women chemotherapy and hormonal therapy, and we're giving it to them to try to minimize the chance that the cancer will come back outside the breast, for instance, in the bones or the liver or the lungs. When the cancer comes back outside the breast, we call that metastatic cancer, and that's what we're trying to prevent. So in this particular study called the Olympia study, which is a international study, the idea is to try to use one of these PARP inhibitors after someone's finished their initial chemotherapy for an early stage breast cancer. And women either are randomly assigned to receive olaparib for a year or a sugar pill, and then you wait and see what happens. So some people say, well, why not give it to everyone? But we don't know yet that about the risk-benefit ratio. Any drug has side effects. Any drug has potential toxicity. And so it's very important to know what those side effects are in this situation, and whether or not we're doing, we want to be doing more good than harm, and we need to specifically look at that closely. So that is another study that we're working on. But ultimately, what we'd like to do is prevent cancer in the first place. And the group on the phone knows very well that right now a lot of the prevention efforts are focused on surgery, prophylactic removal of the ovaries, preventative removal of the breasts. And although these things do significantly decrease the risk of development of cancer, they're not exactly what we are hoping for as the final answer. So we are very dedicated to and devoting to finding other solutions to prevention apart from surgery. Now, one way to do that is to take those medications that I just told you about, and if they are safe and effective in early stage breast cancer, you can then ask the question, oh, could you use these for prevention? But again, you have to do it stepwise. You need to make sure that you're doing more good than harm. Likewise, Dr. Greenberg will tell you about all the ways that we're trying to figure out what these proteins do in the cells. And the more we know about that, the more we understand how these genes and the proteins they make work, the more we're able to figure out what different prevention options are. However, another thing that we're actively working on is to use the immune system, and specifically to think about using a vaccination strategy to prevent BRCA1 and 2 related cancer. So if you had said 10 years ago that we were working on a vaccine to prevent cancer, a lot of people would say we're crazy, and there are probably still people who say that now. But what we've really figured out in the last 5 years is that it's not a pipe dream, that you can use the immune system to help fight cancers. We've shown that definitively in things like lung cancer, melanoma, bladder cancer. It is really a pretty remarkable achievement. So now we just have to turn the immune system on to the concept of preventing these cancers in the first place. So how do we do that? Well, we've worked very closely over a number of years with various groups to really figure out the vaccine technology. It turns out, like much of the science, the devil's in the details. You have to actually have a right vaccine that will actually stimulate the immune system to do what you want. So right now what we're doing is vaccinating individuals who are at high risk of recurrence after their initial therapy. So women with breast cancer who have positive lymph nodes, or individuals who had a pancreatic cancer that was resected, things like that. And they are receiving 4 doses of a vaccine to see whether that vaccine is safe and whether there's been an immune response. So far we've enrolled 57 patients into this study and are working our way through. It's safe. There aren't any issues with that. We're now working our way through the immune responses and seeing whether or not we need to tweak the vaccine to make it even more able to generate an immune system, or whether we're good to go. Once we figure out if the vaccine's safe and generates an adequate immune response, our goal is to take it into women who have BRCA1 and 2 mutations that have not had cancer. And again, we're not ready to do that right this minute, but that absolutely is the goal. If we show that it's safe and generates an immune response in a sort of a smaller subset of women who haven't had cancer but have gene mutations, then the definitive study would be a randomized clinical trial. So we're thinking big. This is where we are with this, and we are determined to move forward. The other really clinically based research that we're doing, and by the way, I'm not covering all the research that we're doing right now at Vassar Center. I'm just trying to pick out some highlights. But we're also looking at ways to expand access for genetic testing and to let more women have the opportunity to know whether they're at risk for BRCA1 and 2 mutations. Some of this is, straightforward. We know that all ovarian cancer patients should have BRCA1 and 2 testing, and we know that once someone's known to have one of those mutations, that it's really important for family members to get tested. But that's not always happening. So we're trying to figure out different ways to get testing done using telemedicine, using digital education. We're trying to figure out how best to help people talk to their family members. How do you encourage that cousin that you haven't seen very often, but you know about your BRCA1 mutation and they're not interested in the information? Well, how do we help you encourage them? We would love to contact them directly, but we can't. So what's the next best step? And we also are doing work on issues related to children. So, for instance, right now we recommend testing in general at around age 25. It can be different depending on family circumstances, but 25 is the usual age for which young women get tested because that's when we start breast MRIs. But we need to figure out better ways to help people talk to their children and figure out the right strategies there. So those are all things that we are working on. In addition, Mindy and John Gray, who were the Bassler Center founders from the beginning, made it clear that they valued collaboration, and the only goal was to accelerate progress in this field. So however we needed to get that done, that was what we wanted to do. So we do give out grants through a Bassler External Grants Program, and we have to date awarded close to $2 million through this program. We have our next rounds of awards that will be announced in the next few weeks. And the way that this works is that we call for proposals, and we try to send this out very broadly through the world. We have many different contexts. We send it to all the medical schools. And then the larger proposals are reviewed by an external review committee, and we're looking for projects that are interdisciplinary, but also some that are high risk and high reward. In the end of the day, we want potential for translation into clinical practice. So our first large team science award was awarded to MD Anderson Cancer Center. It was led by them, but it also had collaborations with Memorial Sloan Kettering and the Bassler Center investigators. And this was funded to study new mechanisms of chemotherapy responsiveness of BRCA-related cancers and also overcoming therapy resistance. Being responsive to chemotherapy and resistant to therapy are sort of two halves of the same coin, and you want to understand both of those. And the aims were obviously to develop more effective treatment strategies. You're going to hear more about these issues from Dr. Greenberg in a minute. In 2014, we also gave out 4 innovation awards, which were $75,000 to $100,000 awards. And you can see that this spanned Hopkins, Columbia, Fox Chase Cancer Center, and Drexel, and it included community outreach projects and also some what we consider very basic science, which is truly mechanistic work, understanding how things work in the cells. In 2015, these were our award winners. Again, Dana-Farber, Boston Children's, Emory University, and a specific study discussing genetic testing in the Orthodox community. As many of you on the call know, individuals of Jewish ancestry have a much higher risk of having a BRCA1 or 2 mutation. So this is an example, and again, our 2016 awards were just figured out at the end of 2016, and it'll be announced in the next week or two. So we're very committed to research both within our institution and also figuring out how to fund work that's outside of our institution. So I'm going to answer just a few questions because questions have already come up before sending this over to Roger. The first one that was asked was about the issue of pancreatic cancer and what surveillance is available. This is an area also of ongoing study. Right now there is no specific guideline regarding what someone should do about pancreatic cancer screening. However, endoscopic ultrasound, which is putting a little camera down into the through the esophagus into the stomach and basically looking from the inside out to the pancreas is something that has been studied and explored. So particularly for individuals who have a BRCA2 mutation and also have a family history of pancreatic cancer, it is something to speak with your physicians about, and it's also a good idea to figure out what kind of pancreatic cancer screening studies may be available. MRI of the abdomen is another possibility, although that can be difficult to have insurance coverage. So we're actively working on this issue, including whether or not blood-based assays could also help early detection of pancreatic cancer. We're a little ways away from that yet, but there's active work going on. Another question. Another issue was related to insurance information, insurance and getting sons tested. And the issue that was brought up was that when this individual's son went to go get testing, that the insurance company didn't seem to cover this. This is a challenge, and it's insurance company to insurance company dependent on what this will be. And it is true that, for instance, the U.S. Preventive Services Task Force doesn't specifically discuss issues related to men. I think many of us are working on this. The fortunate thing is that in the last few years, a number of much cheaper tests have become available to get people the testing that they need. But I agree that this is an issue, and I do assure you that we are working on it. Related to men, someone asked, and I would like to correct this because they're right. They asked that they said that I mentioned women with breast cancer for many of these studies, and that was an oversight. In most of these studies, men who had breast cancer were also eligible, and it's worth reminding ourselves again that although we talk a lot about women with BRCA1 and 2 mutations, men who have particularly BRCA2 mutations are at risk for male breast cancer, they're at risk for pancreatic cancer, and they're at risk for a particularly aggressive form of prostate cancer. And we have studies involving all of those things as well. So we don't always focus on the men, but we should. We did have a webinar, just so you guys know, that's archived specifically on men with BRCA1 and 2 mutations. The question about PARP inhibitors and lubinectin for pancreatic cancer, and yes, these are really good questions. We know that it looks like BRCA1 and BRCA2 related cancers, if they have developed because there's an inherited susceptibility via BRCA1 or 2, it seems like if you have breast cancer or pancreatic cancer or prostate cancer or ovarian cancer, that there's some likelihood that you may respond. So these drugs may be targeted to BRCA1 and 2, not necessarily the specific type of cancer. And so it's why so many people are interested in establishing whether or not there's a specific genetic susceptibility. And I'm going to do one more question and then turn it over to Roger, and then I'll look through the questions and we'll answer more of them at the end. There was a great question about the difference between sporadic and germline BRCA mutations in ovarian cancer. I think it is, I'm going to start with some basics for the group, which is that if you just say women with ovarian cancer, and then you ask the question, what percent of women with ovarian cancer have a germline or inherited? That's what we mean by germline. You were born with a mutation that's in every one of your cells. About 15% of individuals with ovarian cancer have an inherited mutation in BRCA1 or BRCA2. It turns out that some of the ovarian cancers, even if there is not an inherited susceptibility, some ovarian cancers have developed the BRCA1 or 2 mutation in their tumors. And they, too, seem to respond to PARP inhibitors. BRCA1 and BRCA2 mutations that only occur in the tumor and you weren't born with seem to be less common in breast cancer than they are in ovarian cancer. So this is a reason, a potential reason that PARP inhibitors seem to have a more broad effect in ovarian cancer in general, even though the people who respond best have inherited susceptibilities in BRCA1 and 2. So I know that's sort of a lot of information, but I'm going to let Roger now talk more of the basic science, and I'm going to start clustering all of the other questions so that we can get to them at the end. So Roger, with that in mind, I think you can just click on your slides, and we'll get started with you.
Great. Thank you very much, Susan. I just want to make sure I can see my slides here. I'm actually not seeing my slides anymore on here. Yeah, here they are. All right, I see my face, and great. All right. So thank you, everyone, for taking the time to attend this webinar. My name is Roger Greenberg, and I'm a faculty at the University of Pennsylvania and work closely with Dr. Domchek and others at the Vassar Center to understand basic mechanisms of BRCA cellular function and how we can connect their roles in repairing our genomes, in repairing DNA damage to both the genesis of cancer, cancer etiology, and therapy. I'm not seeing the slides advance here.
Did that work? I'll just advance them for you.
Okay, if you could go to the next slide, thank you.
I'm on presentation objectives. Do you see that, Roger? Or?
I do not see that anymore. Previously, I was able to do it, and now I can see that you've advanced them.
Okay, that should work then.
All right, thank you. So there are several objectives right here that I'd like the audience to walk away with. I understand that there's probably a broad range of scientific backgrounds within everyone attending this webinar. And so really what I would like to accomplish is for many of you to gain a general understanding of how BRCA proteins function in cells, how their involvement in DNA repair suppresses cancer and influences response to new therapies, how resistance to specific therapies occurs and why that is such a clinically important problem, and how the Vassar Center is approaching these critical issues that impact patient outcomes. We can go to the next slide. Susan, can you go to the next slide, please?
I did. You're good.
Okay. I guess it takes a second here. Okay, great. This is a picture of DNA double helix that probably most of you have seen something like this before. Our DNA in our human genome, so all of the DNA, is more than 1 trillion base pairs of a code that encodes for all of the proteins that are expressed in our cells, and which specifies why our cells have different functions and so forth. For example, some cells may express different proteins that lead to function in the brain, and others in the skin, others in blood development, and so forth. There are many different exposures that our cells encounter every day that actually result in damage to the DNA. And in fact, it's estimated that there are more than 10,000 damaging events to the DNA, to our DNA every day. There are a number of proteins that are involved in the cell that actually repair this DNA damage, and there are actually many different ones. Susan, if you could click again.
Yep, I did.
Okay, and it should come up in a second. BRCA1 and BRCA2 are just several components of this really vast cellular armamentarium which is involved in repairing our DNA damage. What they do is specifically recognize different types of DNA damage. Here I indicate over DNA double-stranded breaks, which are breaks on the backbone of the DNA on either side of the DNA double helix, and also DNA intrastrand and interstrand crosslinks. Please click again. What BRCA1 and BRCA2 are involved with are what are known as error-free mechanisms of DNA repair known as homologous recombination. And this restores the DNA back to its original state. And what it does is protects our genomes from encountering and accruing different errors that would lead to cancer susceptibility. Now, mutation to either BRCA1 or BRCA2 greatly impairs homologous recombination, and this leads to increased cancer susceptibility. For example, up to an 85% lifetime risk of breast cancer in patients that have BRCA1, inherited BRCA1 or BRCA2 mutations, and 20 to 40% risk of ovarian cancer, as well as increases in other cancers such as pancreatic cancer that Dr. Domchek mentioned in the previous presentation. So I'd like to just go over a few basic facts of BRCA1 and BRCA2 that I think often there's quite a bit of confusion among the lay public. Patients or other individuals will often state that they have the BRCA1 or BRCA2 gene. What they actually mean is that they have a mutation or one defective copy of either BRCA1 or 2. All humans have 2 copies of the BRCA1 and of BRCA2. A single inherited gene mutation to BRCA1 or BRCA2, denoted as BRCA plus minus, with the plus being the normal copy and the minus being the mutated or defective copy, confers high cancer risk. What happens is that the BRCA mutant cancers lose the normal BRCA gene, and this is called loss of heterozygosity. And therefore, what happens is the patient, or every cell in the patient body, is still BRCA plus minus. They have one normal copy that can repair DNA damage. But the tumor, on the other hand, loses the normal copy. And so instead of being BRCA plus minus, it is now BRCA minus minus. This has a number of consequences. One is the patient, every cell in the patient can repair DNA damage normally, but the tumor cannot. And therefore, the tumor is susceptible to chemotherapies or drugs that require homologous recombination or require DNA repair. And thus, this leads to the property of synthetic lethality, which inspired the development of PARP inhibitors, which I'll show you in a minute. Now, if you look, there's a picture here of cells that have been treated with something called ionizing radiation, which breaks the DNA. It creates a break to either strand of the DNA. And you can see on my cartoon on the right sort of a lightning bolt that would break the DNA, and BRCA1 is right there. This picture, this image that I'm showing you, is really the original discovery of how BRCA1 and BRCA2 worked in cells, and it was performed by David Livingston in 1997. If you can click again, Susan. What they did was a very simple experiment where they used an antibody specific to BRCA1 in this. All right, if you click again, it should come up. And that antibody concentrates right where BRCA1 is, all right, showing that it's at the site of DNA damage. I think that animation didn't come up, but that's okay. And it just gives you a sense of how laboratory investigation oriented the entire scientific field to how BRCA1 and 2 functioned in the cell. They responded to DNA damage that led to the identification of additional suppressors of breast and ovarian cancer that are mutated in familial cancer syndromes, and it also inspired the development of specific therapies such as PARP inhibitors and other agents. The next slide is a concept called synthetic lethality, which was originally hypothesized by Nobel laureate Lee Hartwell in 2001. The statement at the top just says that synthetic lethality arises when a combination of mutations in 2 or more genes leads to cell death, but either mutation in these genes would not. And you can see that in the bar graph below. A mutation to either gene A or B, you can see the first is a mutation, the second pair of columns is a mutation to gene B, the cells are alive. Right next to that is a mutation in A, where the bar graph is lower, and A has diminished function, the cells are still alive. But if you go on the far right of this bar graph, and you can see a combination of impaired function of gene A and gene B, and the cells die. So I'd like to put this into the real-world context. On the right, obviously, you see this woman in pink is BRCA plus minus. She has an inherited mutation to BRCA1 or BRCA2 that would render one copy of the gene defective. On the other hand, a tumor that arose in this patient, in either the breast or the ovaries, would have lost the normal copy of BRCA1 or BRCA2 and is minus minus. If we apply this concept of synthetic lethality, for example, by using PARP inhibitors, the tumors would be much more sensitive or would be killed by the PARP inhibitors, whereas the patient would tolerate this as would any other individual. If we go to the next slide. Is that coming up soon? Next slide. I'm sorry, everyone.
I think you're in the audience too, perhaps, but yeah, do you see it now, though? It's the farmer?
Now I do. All right, what I'd like to do with the audience now is really show some data. And I can take you through this. Some of you may be very familiar with looking at, at numbers, and some of you may be less familiar with this. But I'll take you through this slowly, and I'd like to keep it simple. This is original data from landmark studies from 2005 in Nature from two groups, one from Alan Ashworth's group, who was at that time in the UK, and the other from Thomas Halliday's group, who was in Sweden. And what you can see are three different cell types and their response to PARP inhibitors. The black squares represent cells with 2 copies of normal functioning BRCA2. The triangles represent cells that have 1 normal copy of BRCA2 and 1 defective copy. And the black circles represent cells that have 2 defective copies of BRCA2. Unfortunately, my cursor doesn't work to sort of point these out, but if you can look at the cells, at the curve for the black rectangle there, you can see that very low concentrations of these PARP inhibitors are killing these cells. On the y-axis, or the vertical axis, is the surviving fraction of cells. And their concentration that kills about 50% of these cells is at on the bottom, 10 to the minus 7th. On the other hand, if you look at the cells, the triangle or the square, they are much less sensitive. In fact, they are 100 to 1,000-fold less sensitive to these PARP inhibitors. You can see their curves on the right than are the defective copies. This represents this concept of synthetic lethality very vividly. By creating a defective PARP1, by inhibiting PARP enzymes, this kills cells specifically that have 2 BRCA2 mutations, but is not toxic to cells that have 1 or none. And this concept, only 4 years later, was shown in a clinical trial that was published in the New England Journal of Medicine in 2009, which translated this landmark basic science findings by showing proof of concept that patients with BRCA1 or BRCA2 mutations responded to PARP inhibitors. This then led to approval of the first PARP inhibitor in 2014, as Susan mentioned, olaparib, and then later rucaparib in 2016. Next slide, please.
Roger, I think it's mostly delayed for you. Everyone else is seeing them.
All right. Sorry about that. Okay, so we'll go to the next slide. And what you can see here is a pie chart. Okay, what you can see here is a pie chart of ovarian cancer patients and their different mutations. And below that, you can see survival curves. Ovarian cancer represents a very striking example of the importance of the type of DNA repair that BRCA1 and BRCA2 achieve in the cell, and their importance for both the genesis of these cancers, so their cancer etiology, and also their response to therapies. Ovarian cancer is treated with a drug called platinum. There are different types of this platinum drugs. Carboplatin is one of the names, cisplatin the other. And the cellular response to both of these is pretty similar. About 50%, or half of this on the right side of this pie chart, have mutations in BRCA1, BRCA2, or related genes that render these cancers deficient in homologous recombination types of DNA repair. On the left side of this pie chart are another group of ovarian cancers, about 50% or so, that have proficient or normal homologous recombination mechanisms of DNA repair. And this has a tremendous impact on the patient response to therapy. If you look below, here are survival curves. All of these patients have been treated with platinum plus another drug called paclitaxel. In blue are the curves for the BRCA mutant or the homologous recombination deficient cancers. And in pink and gray are the ovarian cancers that perform homologous recombination well. What you can see is there is a vast difference in terms of their response and overall survival. The curves on the right, the patients survive, about 50% of the patients survive to platinum for 4 years or longer, 48 months or longer. However, the patients that have normal homologous recombination have much lower survival and much worse response to therapy. In fact, their survival median is about 24 months or less. And so this brings up a number of points. One is that homologous recombination can be treated with agents, with chemotherapy agents, that create a burden on the cell that requires this type of DNA repair, and this can improve patient outcome. But the very sobering feature of this blue curve that you can see, the blue line here, is while the patients survive longer, they are still succumbing to their disease, and thus resistance is emerging even in the BRCA mutant cancers. Please click again, Susan. And so one of our goals, I lead a group of scientists at Penn, many of whom were not working in this area before, were not working on BRCA mutant cancers, is for us to identify and target mechanisms of resistance to PARP inhibitors and platinum-derived agents in BRCA mutant cancers. The second goal that we have is, could we develop predictive biomarkers of response? Could we understand basic biology to the point where we would be able to derive or develop markers that would be non-invasive and which tell us which patients would respond to which drugs, which patients would respond to PARP inhibitors, which patients would respond to platinum, and which patients may respond to other types of drugs. And I'll tell you about our ongoing efforts to do this. Here's a little bit of a complicated slide that describes mechanisms of how cells respond to PARP inhibitors. When BRCA1 and 2 are functioning normally, what happens is when cells are treated with PARP inhibitors, this traps them on the DNA when the DNA is replicating. In order for cells to divide, the DNA needs to be duplicated, and so there are a number of different proteins which replicate our genomes and double them prior to cell division, and then half of the DNA is spread to one copy of the cell and half to the other. What happens is with PARP inhibitors is it traps the PARP enzyme at this replicating DNA, and this creates a stop sign that then requires BRCA1 and 2 to repair by homologous recombination mechanisms. Click again, Susan. So if you can see the animation that's arising here, what happens when BRCA1 and BRCA2 or BRCA2 are defective in these tumors is that this type of DNA repair cannot occur, and instead error-prone pathways take over, and this leads to cell lethality because of really genomic chaos. The DNA gets scrambled all over. It brings up a number of points. One is it suggests the resistance mechanisms that can arise, and there are a number of them that have been validated and a number that we still don't know about. Some of the resistance mechanisms are that BRCA can be mutated a second time to create a normal copy or a partially functioning copy of BRCA1 and 2 that can do homologous recombination DNA repair in the cancers and make the cells resistant to PARP inhibitors. Another mechanism of resistance is to actually lose the target of the drug. If PARP1 expression is lost, these cells are no longer sensitive to the drug anymore. And finally, error-prone mechanisms of DNA repair can be eliminated by these cancers, and this is a third mechanism. And so our goals are to be able to develop non-invasive biomarkers to predict which of these resistance mechanisms is occurring, or will the cancers be sensitive, and also to develop novel strategies to restore sensitivity to these drugs in BRCA mutant cancers. So let's go to the next slide. All right, if you can see this, one of the things that we've developed is from a radiochemist named Bob Mock, who works in our group, who's developed a version of rucaparib that can be labeled. And you can see at the F atom on this drug, I'm showing the structure of it, it can be labeled with an isotope called fluorine 18. What this allows is a type of imaging called positron emission tomography, which is actually used clinically. And I'll show you some examples of that soon. This drug when it's treated in ovarian cancer cells, you can see on this graph on the far left is this UWB1.289. This is a BRCA1 mutant ovarian cancer line. When we treat these cells with this, they uptake this isotopically labeled fluorine-18 labeled PARP inhibitor. If we put BRCA1, normal BRCA1, back into these cells, so they've restored BRCA1 and now are resistant to PARP inhibitors, they take up less of this. We've genetically engineered these cells using a technique called CRISPR-Cas9, which actually has been highlighted in the New York Times and in the lay press. And what it allows us to do is eliminate this PARP1 or target of this drug. And now these cells no longer uptake this labeled PARP inhibitor. If we look on the right, this correlates very well with our response to PARP inhibitors. And just to simplify this, what you can see is the bar graph that goes in the negative direction in red in this UWB1.289, this BRCA1 mutant cell line. It tells us that these cells are very sensitive to PARP inhibitors. On the other hand, all of the other group are resistant to PARP inhibitors. If you look, the bar graph goes in the positive direction, and they are tenfold or more resistant to this type of agent, meaning that you would have to use tenfold or even up to 100-fold more drug to affect these cells, whereas the BRCA1 mutant on the far left are very sensitive. So the question that we've had, and our goal, is to use this agent to identify patients that are likely to have a favorable response to PARP inhibitors or platinum, and to be able to determine which patients should be treated with these drugs and which should not. Let's go to the next slide. In the next slide, we've treated 17 patients with these agents. All right, this is now an ongoing clinical trial that has several goals in mind. The first goal, if you can see this study, was to see could we actually quantitatively image cancers that were expressing PARP1 versus those that were not in patients with ovarian cancer. The second goal was to see if uptake of this drug would be indicative of which patients were most likely to respond. We've now imaged 17 patients, and we can definitively answer the first question. We can observe, as you can see where these arrows are on the left side of the slide, we can observe uptake of these PARP inhibitors in ovarian cancers and in their metastases. And you can see this lighting up right where the yellow arrow is pointing to. This is patient 7. On surgical specimens from this patient, we can also use these agents to see how much they uptake. And you can see this, what it says in this patient is a glowing sort of vaginal surgical specimen of the cancer. If we look on the graph on the upper right-hand corner of your slide, what we can see is the average intensity of uptake of this labeled PARP inhibitor. And we can see some patients on the far right have very high uptake, indicating that they're strongly expressing PARP1, and the prediction is that these patients would be very sensitive to PARP inhibitors. However, on the left, if you look, these are patients, for example, patient 4, patient 9, patient 15, patient 11. These patients have very low expression of PARP1, and these patients would be predicted to be resistant to the PARP inhibitors. Well, the trial is still ongoing, and we're still discovering and determining what the correlation is between PARP expression in patients versus patient response. The initial results are encouraging that our preclinical studies are accurate, that loss of PARP1 confers resistance to these agents, whereas high expression is indicative of more strong responses. All right, I'd like to continue here. Next slide, please. All right, in the next slide, what you can see is an example of when we restore BRCA1 and BRCA1 mutant ovarian cancers. What you can see on the left is the curve of these cells, BRCA1 mutant cells in black. All right, the black line, these are BRCA1 mutant cells. And at a 34 nanomolar, at a low concentration of olaparib, the PARP inhibitor olaparib, they become very sensitive. If we restore BRCA1 in these patient cells, this results in a shift to the right, a 10-fold or more increase in resistance to these agents. This is a model for what happens in what's thought to be about 30 or 40% of tumors. They have secondary mutations that actually take a defective copy of BRCA1 or 2 in the tumors and convert it to a partially functioning copy, and this makes the patient cancers resistant to therapy. The question that arises from this work is, could we further cripple, in this case, the restored BRCA1 or BRCA2 to make these cells and to make them now, again, sensitive to PARP inhibitors or other drugs? Next slide, please. And so we have a number of strategies to do this in our group, and what we are really looking into is understudied aspects of the field. One of the questions that a member of our team, Dr. Lin Zhang, has approached is, could he find agents that would, at low concentrations, enhance PARP inhibitor responses by crippling this restored BRCA1 or BRCA2? And what he's discovered is a drug called that inhibits a type of enzyme called BET domain or BRD4 inhibitors. These are in clinical trials for other indications, and they are well-tolerated agents that are currently being investigated in a variety of cancers. And so Lin Zhang asked the question, could we repurpose them to restore sensitivity to PARP inhibitors in BRCA1 mutant cells that have become resistant to therapy? What you can see in each well of this plate are purple stain, and this is indicates the number of cells of BRCA mutant cancers that are able to proliferate and grow in the well. So the more purple that you have, the more cells. The wells that are blank indicate that the drug combination is killing these cells. So I'd like to take you to the first two columns in this rectangular plate of cells. What you can see here is in each row they're treated with either a vehicle control that has no effect called DMSO, this drug JQ1, AZD is actually olaparib, or the combination below is the final row. And what you can see is that in the second column going down, that very low concentrations of this JQ1, this drug that inhibits the BET domain proteins, or AZD, the PARP inhibitor, have very little effect on these cells. But if we have the combination in the very last row, so second column, last row, you can see that there are very few cells left on the plate. This low concentration of two drugs has now restored sensitivity to PARP inhibitors. And if we go across the final row here, the fourth row, and go all the way across, we see at every concentration of the drug, we largely eliminate the ability of these cells to proliferate. This is a very exciting result. Dr. Zhang has a number of mechanistic studies that he's performed to understand the basis for this phenomenon, and it's quite interesting. He's submitted these for publication, and we're quite interested in developing these types of combinations for entry into clinical trials in patients that have become resistant to therapy. I'd like to go to the next slide to show you another example. The next slide uses similar methodology, but a different combination of drugs. This work was performed by Vassar-funded investigators at the University of Pennsylvania, Eric Brown and Fiona Simpkins, and has recently been published in Clinical Cancer Research. There is what is known an enzyme known as a protein kinase called ATR. And ATR inhibitors have been developed for a broad variety of cancers, and they have currently entered clinical trials. Doctors Brown and Simpkins asked the question, based on a number of mechanistic studies that they had performed, of whether ATR inhibitors would also synergize with PARP inhibitors and restore sensitivity in patient-derived ovarian cancers that have become resistant. And what you can see, these purple-stained wells, again, are indicative of the ability of cells of these ovarian cancers to proliferate and grow in the presence of drug. And you can see in the combination on the right that there is very little growth of either. There's no uptake of this purple dye. Now, if we look at the graph on the far right, here is tumor volume. What happens is patient-derived ovarian cancer is injected into mice that are immune deficient and can't reject the cancers. And you can see in the solid black line in the circle, the control, these patient ovarian cancers grow rapidly in the mice, and their tumor volume increases over this 7-week period of which the experiment occurred. When Dr. Brown or Simpkins treated these mice with ATR inhibitors, which are in light gray, or PARP inhibitors, which are in solid gray, this resulted in what we call a cytostatic effect. The tumors didn't proliferate, but they didn't shrink either. So that they were clinically significant in these mice. They prevented tumor growth, but they didn't eliminate the tumor. Now, if you look at the dotted line on the bottom with the clear circles, when they combine PARP inhibitors and ATR inhibitors together, what you can see is the tumor starts to shrink. This combination allows lower doses of each agent, was well tolerated by the mice, suggesting that it might have acceptable toxicity in patients, and it actually shrunk tumors. Furthermore, just like the previous slide that I showed you, it actually restored sensitivity in tumors that had become resistant to PARP inhibitors. And so while the tumors evolve in ways and acquire additional mutations that outsmart our initial therapies, I think that basic understanding of this has allowed us to attack and to further outsmart these tumors. So I'd like to, and these types of approaches hopefully will be entering clinical trials in the near future. I'd like to go to the next slide now. There are additional types of approaches that are going on to understand how different types of signatures and things that have, and aspects of tumor biology that have been very poorly studied in the field can uncover novel agents. And so my laboratory is using a technique called CRISPR-Cas9 to screen for new genetic hits that would cause synthetic lethality in BRCA mutant cancers. Our early results are promising. This is just a schematic of our approach. It's not critical that you understand all aspects of this, but what this will hopefully allow is for us to identify new synthetic lethalities beyond PARP inhibitors that would selectively kill BRCA mutant cancers. Let's go to the next slide. We also have a number of experts that we work with who are at the cutting edge of understanding immune checkpoint therapies that have been gained quite a bit of press lately due to their exciting results in patients, for example, that have melanoma and so forth that Dr. Domchek presented in the previous slide. Dr. Andy Min and Wei Tong, Drs. Andy Min and Wei Tong in our group are studying these types of responses, and what they have developed are signatures of which patients can respond to chemotherapy and which patients do not. There is a signature called the interferon responsive gene signature that Dr. Min has pioneered the use of for predicting these patients, and we are both, and which seems to be very prevalent in BRCA1 mutant cancers. And we are learning how to exploit this type of signature in these patients through a variety of different mechanisms. I'd like to go to the next slide, please. So I hope that I've given you a sense of how this is being approached and how basic science has already had such a critical impact on understanding the mechanisms of disease and developing new therapies for them. If we look at this sobering graph on the left again, once just to reiterate, BRCA or an homologous recombination repair deficient cancers respond better to certain types of therapies, and we can exploit this Achilles heel in these cancers to improve patient outcome. Again, the sobering feature of this is that this is not yet curative. And so our mission is to understand how to restore sensitivity to patients that have become resistant to these therapies and to develop predictive biomarkers of which patients will respond. I think I've shown you a few examples of how we've actually achieved some of these goals in early studies, and how they are actually already being introduced into the clinic in patients with BRCA mutations. We will continue this work that we are quite excited about with the goals of further improving patient outcomes. So with that in mind, this finishes my presentation. I'm happy to take any questions.
Well, Roger, we
Yes.
do have one question that I'll ask you. People wanted to talk a little bit more about CRISPR in terms
Yes.
of fixing the mutated gene. Do you want to comment on that, and I can give it another perspective as well?
Sure. CRISPR is an exciting technology that allows one in the laboratory setting to engineer a gene to either make it defective or actually restore it to a normal copy. And so the question is, could we use CRISPR technology to restore the mutated or defective copy of BRCA1 or BRCA2 in patients? I think this is a very challenging goal. One would have to be able to deliver to an adult patient or even to a pediatric patient, would have to deliver CRISPR in a way to reverse this defective copy in every cell. If we think about this a little bit earlier, this could be used, for example, in principle, could be used by taking embryos ex vivo and fixing the defective copy at that stage prior to implantation, reimplantation in a patient. This type of approach has not yet been approved, and there are no clinical trials, to my knowledge, in human beings.
Right, and I want to point out that just related to that, because it's really important, is that right now we do have the opportunity for pre-implantation genetic diagnostics, or PGD, where people can go through IVF, screen the embryos, and then only reimplant the embryos that don't have the BRCA1 or 2 mutation. And so, until at the embryonic stage, it's not even one cell, right? You're already dealing with a multicellular embryo. So how CRISPR would work in that, it's still very experimental, just to emphasize. But PGD is something that we already do, which is selecting embryos without the gene mutation. Obviously, only if patients are interested, and people have a lot of feelings about that one way or the other.
I would completely agree with what Dr. Domchek just said, and I'd like to add an additional note of caution to CRISPR. CRISPR can create mutations to restore a defective copy of a gene in a laboratory setting. However, CRISPR also has off-target effects where it can create additional mutations either to that gene or other genes. And so it is right now really just used in the laboratory setting, and there are quite a bit of reasons to believe that it could have deleterious effects beyond that one would not want to introduce certainly into a human being. And so the option that Dr. Domchek mentioned about pre-implantation screening, it would be a much more realistic and safe way of achieving this goal.
Another question that was asked is related to the issue about ovarian cancer beginning in the fallopian tubes. We happen to have an expert at that at Penn, Ronnie Drapkin, who did a lot of the initial studies related to this. And Roger, do you want to give a research answer, and then I can give a more clinical answer?
Sure. I can give a little bit of very brief history of this. This was first observed by a Dutch scientist on patient specimens that early lesions appeared to be in the fallopian tubes, and quite a bit of work done by Chris Crum and also by Dr. Chris Crum at Brigham and Women's Hospital and by Dr. Drapkin at the University of Pennsylvania showed that many of the early lesions are in the fallopian tube, and particularly in BRCA mutant patients. This has a number of potential implications in terms of prophylactic surgery and so forth, and sort of makes us re-examine some of the early assumptions. A model of BRCA mutant ovarian cancer was created by Dr. Drabkin at the University of Pennsylvania, in which he can actually recapitulate much of the ovarian cancer sort of etiology in mice by targeting BRCA and other mutations to the fallopian tubes. So, Dr. Domchek, if you want to
right.
comment on that.
The question that gets asked clinically, and importantly so, is, hey, if I could just take out my fallopian tubes and leave my ovaries in place, well, that's like having your tubes tied. That doesn't take away your ovarian function. It doesn't take away your estrogen. And so, although there's a lot of interest in this approach, it's also important to realize that we don't yet know when those cells in the fallopian tubes, if you will, go bad and when they leave the fallopian tube. So the current standard of care still is to remove the ovaries, but we are actively investigating this because obviously if we could leave the ovaries in place for longer, we would definitely want to. So it's still considered investigational, although there's a number of different types of research that are trying to address that very, very important question. I have another question, which is, do we know if mutations in other genes lead to sensitivity to some of the drugs that we talked about? And this is just worth a brief overview that in people who have inherited susceptibility to breast and ovarian cancer, it remains true that the most common inherited susceptibility genes are BRCA1 and 2. However, we have learned about other genes where if you inherit a mutation in one of these other genes, there's also an increased risk. And interestingly enough, a lot of those genes are in the pathways that Dr. Greenberg just described. They're all kind of clustered together. They're not the same kind of syndromes. A lot of these genes don't have nearly the same kind of risks associated with them with BRCA1 and BRCA2. And there is some suggestion that some of these genes, but not all, but some of these genes may have the same types of responses to PARP inhibitors. So it's early days and all that. These are actually, mutations in these other genes are actually much less common than BRCA1 and 2, and so the appropriate clinical studies in terms of responsiveness to certain targeted agents has not been completed, but that work is well underway, and there are some data that are evolving in ovarian cancer. So, Roger, I don't know if you want to add anything.
I think that was very well worded, and I think what it also highlights is the importance of comparing the basic science studies to the clinical studies. Many of these genes that are mutated at lower frequencies in familial breast and ovarian cancer that are not due to BRCA1 or BRCA2 mutations, many of them were discovered by doing some of the basic science to understand how BRCA1 and BRCA2 worked. For example, there's a protein called PALB2, and that acts as a molecular linker between BRCA1 and BRCA2. BRCA2 binds to it, PALB2 then binds to BRCA1, and it connects them all in what we call a complex, or it's really working like a machine. And if you create or mutate PALB2 in cells or BRCA1 or BRCA2, there are similar responses to PARP inhibitors and so forth. The real test, though, and these genes were then, many of them were found to be mutated in familial breast and ovarian cancer syndromes. The real test, though, as Dr. Domchek mentioned, is what is the risk that mutations in these genes actually cause for these patients, and how do their tumors actually respond to PARP inhibitors? And I think that data is still emerging, and it will be quite interesting to understand the relationship between the preclinical basic studies in the laboratory and what happens with patient response. And ultimately, that will direct the clinical treatment, and it will also inform the basic scientists about the sort of inpatient relevance of our preclinical laboratory findings.
And another question is related to whether or not we have better, new, appropriate tests for the early detection of ovarian cancer. again, to put this in context, ovarian cancer detection has been notoriously difficult. To put it in contrast, breast cancer, particularly breast MRI, is very good at detecting early-stage breast cancers, and most women with early-stage breast cancer do very well. With ovarian cancer, most women who are diagnosed with ovarian cancer are diagnosed at a later stage where their prognosis is not what we want it to be. So it's been really the holy grail to look for new ovarian cancer screening tests. One of the challenges is that these tests are difficult to come up with. So over the years, there have been a few different iterations which haven't really passed the highest bar for a good screening test. More recently, there have been some studies looking at something called a ROCA algorithm, with more frequent CA-125s, but that still didn't pass the appropriate bar for us to really recommend that as a general screening test with great confidence. There are sort of some novel tests that are being done, if you will, using almost kind of a special pap smear to see if there can be shed ovarian cells that we can look for, whether or not we can come up with new biomarkers in the blood. I recognize that people can be frustrated with the fact that we've been working on this for so long and we don't have a good test, but it does show how difficult it is, and people are highly motivated. So hang in there. We're still working on it, but we really haven't gotten to the point yet where we feel that there's an appropriate test that can substitute for oophorectomy. Roger, any further comments on that?
This is really a very, very important problem, and no other comments. This is an active area of investigation at the Vassar Center and beyond.
Someone lost their sound. Sorry about that. I'm looking at all the questions. Another question was if you've had a mastectomy and you've had your ovaries removed as well, what do you need to do in terms of surveillance? And one thing that we didn't really touch on in this particular talk, although we've talked about it in other webinars, is that we talk about BRCA1 and BRCA2 as if they're equal. And BRCA1 and 2 are related, but they lead to different cancer susceptibility syndromes. What do I mean by that? Well, BRCA1-related breast cancers are generally what we call triple negative. They do not express estrogen receptors on their cell surface. The risk of ovarian cancer is higher in BRCA1, and yet the risk of other cancers seems to be somewhat lower. In contrast, BRCA2 mutation carriers, the women have lower risks of ovarian cancer, but there seem to be risks for melanoma. There's the increased risk of pancreatic cancer that we talked about, and there's also this increased risk of prostate cancer that seems very clear-cut for BRCA2 and less clear-cut for BRCA1. And so if you have a BRCA1 mutation and you've had your ovaries removed and your breast removed, in general, we recommend chest wall exams, just a physical exam, monitoring lumps and bumps in your breast, and including a chest wall exam by a physician, and periodic pelvic exams, but mostly just attention to symptoms. Fortunately, the risk of developing ovarian cancer after you've had your ovaries removed is low. It's not zero, though, but it is low. And then for BRCA2, we actually at Penn do recommend at least a baseline dermatology exam, and then if you do have a lot of moles, potentially to be seen by dermatology yearly. We talk about pancreatic cancer screening trials, and for the men, we also make sure about PSA screening, prostate cancer screening, because although there's controversy in the general population, although there's controversy about PSA screening in the general population, we do recommend it for our BRCA1 and BRCA2 mutation carriers. Another participant asked about research opportunities, and we're always very excited about people being interested in research. On our website, vassar.org, there's possibilities of connecting with us, but we'll reach out to you directly, the person who asked that question. There's a lot of different ways that people can participate in research. And as a reminder, the goal of the research is to answer questions that are important to you as well. So please let us know what they are. We also have an additional question, which is more complicated, but I'll try to go for it. Which is, in the Ashkenazi community, Ashkenazi Jewish community, in addition to the fact that there are increased risks of having BRCA1 and BRCA2 mutations, there are also other genetic risks, and one which was raised was a genetic susceptibility to Parkinson's disease. We actually don't know of any association between those two, at least that I'm aware of, but I'll be sure to sort of investigate further. And so with that, we have about 4 minutes left, and so Roger, I'm going to give you an opportunity to, I'm just checking, we got a flood of last-minute questions. This is a good question. So the question was whether or not individuals who have both a BRCA1 and a BRCA2 mutation have different cancer risks. And so before you wonder, could that actually really happen? Yes, it does happen. It happens more commonly in individuals who are of Jewish descent because these mutations are so much more common in individuals who are of Jewish descent. And it's actually a reason why if we are testing someone whose parent has a mutation in BRCA1 or 2 and they are Jewish, we always test for the 3 common Jewish founder mutations. We don't just test for that no mutation in the family because this can happen. In the non-Jewish population, it's quite rare, so unless there's a strong family history on the other side, it's not something that we really look for. But in the rare patient that has both a mutation in BRCA1 and BRCA2, it doesn't double the risk, if you will. What it looks like is that you get those risks associated with BRCA1, so more of the triple-negative breast cancers and the higher risk of ovarian cancer that's consistent with a BRCA1 mutation carrier. But you also have the risks associated with a BRCA2 mutation, which are an increased risk, for instance, of male breast cancer, the prostate cancer if you're a man, and the increase in pancreatic cancer. So if you will, you get the risks of both, but you don't have a higher risk of breast cancer or a higher risk of ovarian cancer. And this is actually something that has been directly looked at. There is a very large consortium called SIMBA, and this consortium is an international consortium that many, many centers contribute to. So you can feel good about the fact that in this field, the researchers are very highly collaborative, and the only way that we could look at that question is because there are over 40,000 BRCA1 and BRCA2 mutation carriers who around the world have contributed data and DNA so that we could answer these important questions. And it's really a testament to you all who are interested and supportive of research so that we can make sure to look at these important questions. So, Roger, any final thoughts in your last 30 seconds?
No, I'd like to thank everyone again, and I'm happy to take questions in the future from people if they have additional ones. I understand that not everyone has a basic science background, but I hope that you gained some appreciation of what we do in the lab.
Terrific. Well, thank you, Roger, for your participation. And with that, we'll thank you all, and we look forward to our next webinar, which we will announce shortly. Sam, any other words for closing?
Just if you want to listen again or you want to recommend the webinar, it will be online later this week, and there will be a live link in our archive. And thanks, everyone, so much for listening.
Great. Thanks again. Have a good night. Bye-bye.